Welcome back, students. Today we are going to discuss part 9 of Unit 2, that is Unit of Life. So, before I start the main lecture, I would like to inform that images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT or have any concern, any issue, please contact us at sathee@iitkey.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss process of mitosis. Okay, process of mitosis. So in last lecture, in part 8, we have discussed different phases of cell cycle, right? G1, S and G2. These are the phase. To collectively we can use the term interphase for the same. Interphase. That's why it is written over here, interphase. Okay, so during interphase, cell prepare itself for the mitotic division. Okay, so as I told you, in the G1 phase, cell prepare different type of biomolecules. Okay, prepare and synthesize different type of biomolecules. That's why it is written over here, cell growth occur in G1 phase. In S phase, DNA synthesis is going on, right? So duplication of chromosome occur in this particular phase, or synthetic phase. That's why we can use the term S or synthetic phase for this phase where DNA synthesis occur, right? And in G2, RNA transcription, right? Translation and duplication of some essential organelles occur in this particular phase. Okay, and esterase preparation, spindle fiber formation, this is also another part which also develop in this particular phase. And after G2 phase, cell come under this mitotic division phase. And this mitotic division phase we can further divide into two different type: mitosis or meiosis if gametes are involved, and cytokinesis, or we can say karyokinesis and cytokinesis. And after cytokinesis, from one parent cell, two daughter cell, formation of two daughter cell occur. And the total time of this interphase, what is the total length of total time duration? 10 to 30 hour. Okay, 10 to 30 hour whole interphase, right? So this is all about the interphase and their subheadings G1, S and G2. Now let's discuss next heading, that is mitotic phase. Okay, so interphase is followed by mitotic phase, right? During mitotic phase, already duplicated chromosome are equally distributed to the daughter cell. This is the main phenomena of mitotic phase. Already duplication occur during S phase, so equal division or equal distribution of this duplicated chromosomes occur in two daughter cell occur during mitotic phase. Okay, and this duplicated chromosome contains exactly same hereditary information as the parent cell have. Okay, though the other cell components like organelles, different molecules are also divided approximately equal number into daughter cells, but not as precise as DNA. DNA is distribute itself in a very precise manner. Okay, that's why the offspring, we can use the term offspring are genetically morphologically identical to the parental cell. Okay, so after mitosis is over, daughter cell again further enter into the G1 phase for its growth or for next cell cycle, right? Okay, so this process was first described by Eduard Strasburger, but the term, this mitosis term, has been given by Fleming, Walter Fleming, in 1882. Okay, so the process has been described by Eduard Strasburger in 1875, but the term was given by Fleming. Mitosis. Okay, it is the most common method of cell division in eukaryotes that takes place where? In somatic cells. That's why we can also use the term somatic division for the same. We can also use this term for mitosis, somatic cell division, right? However, in gonads also, it occur in undifferentiated germ cells. Undifferentiated germ cells also continuously divide with somatic cell division. In plant cells, this mitosis is a very common phenomena. All the cells of meristematic tissue is divided through mitosis only. And the duration of mitosis is from 30 minutes to 3 hours. Average 30 minutes to 3 hours maximum. This is the time duration of mitosis. Clear? Sometime question asked on this particular thing, what is the time duration of mitosis? So minimum 30 minutes, maximum 3 hours. Okay, is it clear? Can I move to the next slide? Okay, now mitosis. Now let's discuss, let's see the definition. Okay, so mitosis is defined as the division of parent cell into two identical daughter cell, each with a nucleus having same amount of DNA, the same number and kind of DNA, and same hereditary instruction as the parent cell. Okay, so mitosis is a type of division of a parent cell into two identical daughter cell, right? Each with the nucleus having same amount of DNA, same and same number of chromosome. Obviously, same number of chromosome also there must be there, and same hereditary instruction as the parent cell. So it is 100% identical to their parent cell. So that's why we can also use the term equational division for mitosis. This is another term. Okay, equational division. There are two main events involved in mitosis. First is division of nucleus, or we can say karyokinesis, and then division of cytoplasm, or we can say cytokinesis. These two main events occur in this particular mitosis. Okay, now let's discuss all the events involved in this karyokinesis, as it is a complex process, right? So we can further divide this karyokinesis into 4 stages. First one is prophase, then followed by metaphase, then anaphase, and the last one is telophase. Okay, prophase, metaphase, anaphase, and telophase. Okay, now as there I have put this particular diagram over here through which you can understand better all the phases. This is prophase. First one is prophase of mitosis or somatic cell division. Here you can see nuclear envelope is in intact form. This chromosome start condensing with 2 chromatids, you can see over here. Okay, and these 2 chromatids held together at the kinetochore, this middle part, there is a plate-like structure known as kinetochore. Okay, this is the cytoplasm, and spindle fiber development of bipolar spindle fiber also starts in this particular prophase, right? And after prophase, prometaphase, this is the prometaphase stage where spindle fiber microtubules, development of microtubules occur, and here you can see there is no intact nuclear envelope. This is disappeared, right? Over here, kinetochore microtubules, these are attached through this, and in metaphase all the chromosomes are present over here in equatorial plate on the center, align on a center, right? So this is how at this time we can use the term stationary chromosome aligned to the equator of spindle, right? And after metaphase, anaphase occur. In anaphase, separation of chromatids. Okay, these chromatids being pulled towards the pole with the help of spindle fibers and shortening of kinetochore microfiber. Okay, and then after anaphase, telophase is the last stage of karyokinesis, where again generation of nuclear envelope starts. So nuclear envelope reform, you can see over here like this around individual chromosome. Okay, and then after telophase, cell come into the next phase, that is cytokinesis, means division of cytoplasm occur in this particular phase. So how? Through contractile ring or this contractile ring creating cleavage furrow. This is how one cell, first nuclear division occur, equal division, then cytoplasm, division of cytoplasm occur. So these are the stages of mitosis division or somatic cell division. Now we will discuss each phase one by one. Okay, so this is the stage of prophase 1. Okay, so in prophase 1, thickening and coiling of chromosomes occur. Thickening. Otherwise, in a normal cell, chromosomes are present in a fiber-like structure, in chromatin form, just like a thread. Okay, but as in a very thread phase, and from that chromatin to chromosome phase, they come into, start thickening and coiling of chromosome. It starts in prophase. Okay, so chromatin gradually condense into well-defined chromosome. So initially these are present in chromatin form. Like if this is a cell, this is a nucleus, so like this only. Like this only. This is chromatin form. But as cell enter into M phase and prophase of the M phase, then chromatin gradually condense into well-defined chromosome, as you can see over here, and each consisting of two identical sister chromatids. You can see over here, and each chromosome is having two identical sister chromatin. We can see over here. Nucleolus, a rounded structure within the nucleus, shrink. Nucleus will start shrink and disappear slowly, and similarly this nuclear membrane also start disappearing. Okay, so nucleolus as well as nuclear membrane both start disappearing. Okay, and at the end of prophase is marked by the group of microtubules. Microtubules start to form spindle fiber, as you can see over here. Okay, and chromatid will attach to these spindles and pull apart, ensuring that each daughter cell has the identical and complete set of chromosome. The prophase is a long phase and it is a complex phase that lasts for 50 minutes. So what is the time duration of prophase? 50 minutes. Okay, now metaphase. Next stage is metaphase, and this particular metaphase, what will happen? All the chromosome align at metaphase plate. This is known as equatorial plane or metaphase plate. Okay, this is not a physical structure, it is just a term for the plane where chromosome align on equatorial plane or metaphase plane. Okay, at this stage, two kinetochore of each chromosome should be attached to microtubules from opposite spindle pole, as you can see over here. These attached to this pole, and this another one attached to the opposite pole like this. Okay, so two kinetochore of each chromosome attached to the opposite pole via microtubules, and all the chromosome align at the metaphasic plate. Clear? Now, next phase is anaphase, right? In anaphase, the sister chromatids separate from each other. You can see over here. These attach to the microtubules. So the sister chromatid, this one and this one, sister chromatid, right? So in anaphase, the sister chromatids separate to each other and pull towards the opposite end of the cell, as you can see in this particular picture. And the protein, or we can say glue, that hold the sister chromatid together, that protein is broken down, and that is why now they are not united. They can now separate out to each other, allowing them to this breaking or broken down as this protein or glue that actually holds sister chromatid now is broken down, and this process further allow both the sister chromatid to get separate. Okay, each of now its own chromosome. Now for this, both of this sister chromatid, now we can use the term its own, individual chromosome. So the chromosome of each pair are pulled together opposite end of the cell. Microtubules not attached to the chromosome, elongated and pushed apart, separating the poles and making the cell longer. You can see here comparatively uh, this rounded structure is comparatively longer one, like this in anaphase. Okay, clear? Now next one is the telophase. During telophase, the effects of prophase, prometaphase means disappearance of nucleolus, nuclear membrane are reversed. So telophase, we can say it is just reverse of prophase. Okay, so reappear, as you can see over here, this nuclear membrane again reappear on both the sides, on both the cells, that will, divide very soon. So we can say this telophase is just reverse of, prophase. Okay, prophase and prometaphase, as the chromosome reach to the cell pole. Yes, this reach to the cell pole. We can say see this thing in this particular diagram. This nuclear envelope is again reassembled. Yes, reassembled nuclear envelope, right? Reassemble around each set of chromatid, and nucleoli again reappear. Okay, and chromosome begin to decondense, means chromosome start decondensing itself, convert it into again in chromatin form, right? Decondense back to its extended chromatin that is present during interphase, right? So this phase is just we can say just opposite to the prophase. Okay, so nucleoli reappear, nuclear membrane reappear, develop, or we can say develop again condensed chromosome, converted it into chromatin. So this all events just opposite to, we can say these all events just opposite to prophase. So mitotic spindle is disassembled, and remaining spindle microtubules are also depolarized them. So telophase account approximately 2% of the cell duration. Okay, and after this cytokinesis starts. Cytokinesis means division of cytoplasm, right? So and this in cytokinesis, it encloses the daughter nuclei formed by the karyokinesis in separate cells, thus completing the process of cell division. And the cytokinesis is signaled at the metaphase by cytoplasmic movement that bring equal distribution of mitochondria and other cell organelles into two halves of the cell. So equal division of cell organelles occur during cytokinesis, not during karyokinesis. Obviously, in karyokinesis only equal division of DNA occur, right? So just keep this thing in mind, that all the uh, organelles like mitochondria, Golgi complex, this equal division occur during cytokinesis. Now let's discuss significance of mitosis. First, maintenance of size. Mitotic helps in maintaining size of a cell. A cell with when full grow divide by mitosis instead growing further. So maintenance of size, because continuous growth is going on inside a cell. So after some time that particular cell can increase in the size. So at that time the cell division occur, and again it will get back to their original size. So maintenance of size, first significance. Second, growth. After fertilization, a fertilized egg develop into embryo and finally into adult with this mitotic division only, by repeated mitotic division only. Okay. Third, maintenance of chromosome number, right? So mitosis keeps the number of chromosome equal in all the sets of an individual. Thus, mitosis provide a complete set of genetic information in each cell, since DNA duplication in S phase prior to mitosis. Okay, so this is the third significance. First one is maintenance of size, maintenance of growth, maintenance of chromosome number, repair. Mitosis provide new cells to replace the old or aged cell or dying cell, right? Healing and regeneration. So mitosis also provide new cells for healing of wound and regeneration. Just we have already discussed this with the example of a lizard, with the example of liver, right? Reproduction. Mitosis bring about multiplication of so many asexual organisms. In multicellular organism also, it play an important role in reproduction, asexual and sexual. Okay, evidence of basic relationship of organism. Mitosis being essentially similar to many kind of organisms support the basic relationship of living things. Okay, so this is all about the mitosis, different stages of mitosis. I hope all the concepts related to this topic has been cleared now. Still, if you feel you are having any doubt, any question, you can write down to me in comment box. We will discuss it later on. Till then, take care. Bye bye. Namaskar.